What if we could not only see individual molecules, but also control and probe their electronic states while immersed in a liquid environment that mimics real-world chemistry? This is precisely the promise of electrochemical scanning, tunneling, microscopy, ECSTM. These techniques merge the atomic resolution of scanning, tunneling, microscopy with the dynamic control of electrochemistry, opening a window into the nanoscale world where chemistry, physics, and electronics converge. Let's begin with the basics. What is ECSTM? Standard Scanning Tunneling Microscopy, ST, allows us to image conductive surfaces with atomic resolution by placing a sharp metallic tip near a sample and measuring the quantum tunneling current that flows between them. But this technique usually operates in vacuum or inert environments. ECST, in contrast, works in electrochemical solutions, allowing scientists to study how voltage, ions, and chemical reactions affect molecular surfaces and individual molecules. Why is this important? Because most chemistry in the real world happens in liquid environments like biological cells, batteries, or corrosion processes. ECSTM bridges the gap between solid-state nanotechnology and real-world chemical systems. It allows researchers to tune the electrochemical potential of the tip or sample and watch in real time how a single molecule responds. Does it gain an electron, change its shape, or bind to something new? Consider this question. What happens when you add or remove electrons from a single molecule? Using ECSTM/STS, scientists can measure how redox states, oxidation and reduction, affect the molecule's behavior. These techniques have been used to study electric catalysts at the atomic level, such as single PT or auto atoms in fuel cells, redox-active molecules like porphyrins, which mimic the core of hemoglobin and chlorophyll, organic semiconductors in flexible electronics where switching behavior can be tuned electrochemically. This goes beyond imaging. With electrochemical scanning, tunneling, spectroscopy, ECSTS, researchers apply a range of bias voltages and map out the density of electronic states, DOS, revealing how a molecule conducts electricity, responds to charge, or undergoes conformational changes. You can literally watch molecular orbitals appear or disappear depending on the applied potential. Let us ask a deeper question. Can this be used for energy or medical applications? Yes. One frontier is molecular scale batteries. Researchers are investigating redox and active molecules that can reversibly store energy. ECSTM helps identify how these molecules degrade over many charge/discharge cycles, leading to better design for long-term stability. In biophysics, ECSTM has been used to probe DNA and protein molecules, observing how they fold, unfold, or bind ions and drugs. For instance, single electron transfer events in cytochrome proteins can be tracked as they mimic electron transport in mitochondria. Another fascinating application is in molecular electronics. By using ECSTM to trap and manipulate single molecules between electrodes, scientists can build switches, memory elements, or logic gates, all from individual molecules. The electrochemical control adds a new dimension. Molecules can now be tuned dynamically, giving rise to reconfigurable nanoscale devices. One striking example, in a 2019 study, researchers used ECSTM to watch a single C60 fullerene change its electronic state depending on its redox charge. This demonstrated how quantized conductance, a quantum mechanical property, can be modulated by electrochemical inputs, paving the way for quantum molecular circuits. But what are the challenges? The resolution and stability required for imaging in liquid is extremely high. Vibrations, noise, and molecular motion can make interpretation tricky. Additionally, STM only works for conductive or semiconductive surfaces, limiting the types of samples that can be studied. Nevertheless, continuous advances in tip functionalization, feedback control, and machine learning algorithms for interpreting spectroscopy data are pushing the field forward rapidly. So where's this heading? In the coming years, expect ECSTM/STS to drive discoveries in electrochemical catalysis for green energy, EGT reduction, water splitting, nanobiosensing, where individual molecules are detected via redox or tunneling changes, quantum nanotechnology, where redox control becomes part of quantum gate design, electrochemical AI materials, where switching and memory can be encoded in electro-responsive molecules. Imagine a future where molecular machines swim in electrolytic baths, executing logic or catalysis tasks while being monitored and reprogrammed via ECSTM. Would you like to explore a list of groundbreaking experiments in ECSTM that visualize redox chemistry or molecular machines in real time? Electrochemical scanning tunneling microscopy ECSTM combines atomic scale resolution with precise electrochemical control, enabling real-time imaging of surfaces and molecules under liquid conditions. Unlike conventional STM, ECSTM uses a 3-electrode cell and can vary the electrode potential to observe redox-dependent phenomena. This allows researchers to study dynamic molecular processes such as adsorption, desorption, and conformational switching. Complementarily, electrochemical scanning tunneling spectroscopy (ECSTS) measures local density of states as a function of bias voltage and potential. Together, these tools reveal how electronic properties of single molecules change under controlled redox environments. ECSTM has visualized the structural switching of biological molecules as they undergo electrochemical reduction. Like having a molecular camera that not only zooms in to see individual atoms, but also watches them change color when you turn a knob. The tunneling current in STM is highly sensitive to the spatial distribution of electronic orbitals near the Fermi level. As the biased voltage is swept, electrons tunnel into or out of occupied or unoccupied molecular orbitals, allowing orbital mapping. In ECSTS, this information is obtained while controlling the electrochemical potential, making it possible to probe redox-active molecular states. Such measurements reveal how oxidation or reduction modifies the molecule's local electronic structure. The results can be used to identify frontier orbitals, HOMO, LUMO, and charge transfer pathways at the molecule-electrode interface. Spectroscopy on porphyrin molecules showed sharp changes in the tunneling spectrum corresponding to reversible redox transitions, like reading a building's blueprint by shining light through different windows and observing how the interior glows or dims. Redox reactions are central to energy storage, catalysis, and signal transduction. ECSTM allows monitoring of single-molecule redox transitions in situ, including structural and electronic responses. Molecules such as ferrocene derivatives exhibit clear electrochemical switching that can be correlated with tunneling behavior. These measurements provide insights into Marcus theory parameters like reorganization energy and electronic coupling. Observing electron transfer at this level bridges quantum chemical theory and experimental electrochemistry. Single molecule reduction of ferrocene showed changes in topography and conductance near the redox potential, like watching a car's headlights dim as it switches to battery power, indicating an internal electronic shift. The ability of ECSTM to operate in liquid environments allows direct imaging of conformational changes and adsorption geometry of molecules on electrodes. Adsorbates can reorient, tilt, or flatten depending on the applied potential, solvent environment, and molecular interactions. These changes influence electron tunneling and can be controlled to study molecular switching or logic gates. In electrochemical conditions, hydrogen bonding, ion pairing, and hydration shells play roles in determining surface behavior. Such studies support the development of responsive molecular electronics and sensors. Azobenzene molecules were observed to undergo cis-trans isomerization, driven by electrochemical potential, like a book cover that flips open or closed depending on the humidity and light around it. ECSTS enables the measurement of conductance through individual molecules as a function of redox state and chemical environment. This allows characterization of molecular diodes, transistors, and switches with atomistic detail. By tuning the potential, researchers can modulate electron transmission pathways through different molecular orbitals. This is key for understanding quantum interference, tunneling decay, and charge injection mechanisms in molecular devices. The method is also used to evaluate anchor group performance and contact geometry. Measurements on oligophenylene wires showed conductance switching behavior tied to redox modulation, like turning a faucet handle to control water flow through a pipe, except the pipe is a single molecule. Molecular electronic properties are highly sensitive to their surroundings, substrate, solvents, counterism, and adjacent molecules. ECSTM/STS allows high-resolution mapping of these influences by correlating local topography with spectroscopy data. This provides insights into charge screening, local dielectric effects, and surface dipoles. For instance, different crystallographic orientations of metal electrodes can lead to variations in charge localization and hybridization. Understanding such variations is essential for improving electrode design in energy and sensing applications. A study of cobalt phthalocyanine showed strong site-dependent conductance differences on O111, like hearing how the same musical note sounds different depending on the room's acoustics. ECSTM/STS offers a window into the early stages of catalysis at active sites with atomic resolution. It can identify reactive intermediates, monitor catalyst restructuring, and correlate structure with reactivity under working conditions. In electrocatalysis, EGS, hydrogen evolution, CO2 reduction, such insights help tailor molecular catalysts and supports. These methods also aid the design of molecular junctions for solar energy conversion and nanobatteries. Packing catalytic steps one electron and one molecule at a time leads to precision-driven catalyst design. Visualization of CO binding and desorption on PT111 electrodes provided mechanistic clues for CO2 electroreduction, like filming a chef add each spice to a dish under a microscope and tasting the effect in real time. Performing ECSTM/STS is technically demanding due to the requirement for stable tunneling conditions in electrochemical environments. Vibration isolation, drift correction, and reference electrode stability are essential for atomic resolution. Advanced systems incorporate feedback loops, current imaging tunneling, spectroscopies, CSCs, and photon-assisted tunneling modules. Tip functionalization is another frontier, allowing chemical specificity and single-bond resolution. Miniaturization and automation are making these tools more accessible for interdisciplinary laboratories. Commercial ECSTM setups now offer environmental control chambers to enable long-term in situ studies under physiological conditions, like balancing a pencil on its tip while filming a time-lapse of it changing color under different weather. As ECSTM/STS advances, integration with quantum optics, spin-polarized STM, and ultra-fast techniques will reveal new dimensions of molecular behavior. Researchers aim to map entanglement, coherence, and charge spin separation at the single molecule scale. Machine learning is being applied to analyze vast data sets of tunneling spectra for pattern recognition and material discovery. There is growing interest in exploring biomolecular electron transport, redox enzymes, and DNA conductivity in real-time electrochemical environments. Ultimately, this technology may help connect quantum chemistry, nanofabrication, and synthetic biology at an unprecedented level. Recent studies on photosystem I proteins immobilized on electrodes demonstrate quantum coherent charge transport under ECSTM, like decoding Morse code sent by individual atoms and learning that they speak the language of electrons and photons.